Today, our topic is thrombocytes, hemostasis. Part of hemostasis will be taught today because hemostasis, it will involve two processes. One is formation of platelet plugs, then formation of clot. From today's discussion, you have to learn thrombocytes, how they are formed, when the count increases, what it is called, when count decreases, what it is called. What is thrombocytosis, thrombocytopenia, what are the conditions responsible for this. And you have to know their functions. And electron microscopic structure of thrombocytes, platelets. If you study the electron microscopic structure, what you find within the cytoplasm and on the cell membrane, because the contents of the cytoplasm and the cell membrane is associated with the functions of the platelets, and you have to know it. If you know this, then you will be able to know how actually hemostasis, the process of hemostasis begins. Hemostasis means when there is a cut, injury of blood vessels, blood starts coming out, blood oozes, and within short period of time, there is no bleeding. Bleeding stops, ceases. And the process by which it happened, that is hemostasis. There is another term. Don't confuse this term with that one. That one is homeostasis. Completely different. And this is hemostasis. I will teach you what is hemostasis, what is its purpose. Main purpose is to control all the body functions, so that if there is any enhancement, so immediately it will be brought down. If there is lowering of functions, it will be raised. So the process by which it is done, this is called homeostasis. So this is hemostasis. Hem means blood. Thrombocytes. There are two terms, thrombo and cytes. Cytes we know cell, and thrombo means clot. Cytes mean cell. Clot forming cell are thrombocytes. And how the term platelet came? It's like piglet. Piglet means छोटा बच्चा or pig. Platelets, छोटा cell, small cell. That's why it is called platelets. Platelets. platelets are first thing anucleated, no nucleus, without nucleus. Remember this. These cells are anucleated. So this is not a typical cell. We know a typical cell must have a nucleus, but here you don't have. Similarly, RBC is also having nucleus, isn't it? Yes, she is telling yes, RBC is having nucleus. No, don't note like this. And its size very small, as I said. How much? 2 to 5 micron. And normally RBCs are more than 10 are 7.2. And WBCs are more than 10, more than 12. And monocytes, how much? It was yesterday only. Forgot. So there may be, MCQ. So you have to learn. So, yesterday somebody was telling, sir, I don't understand how to prepare. Whatever I am talking about, so you have to prepare that. So all are important. So these points are also important. What? The half-life of thrombocytes. How many days? 10 to 12 days. So they may survive for 20 days also. It depends. It depends. If there is very need, urgently required, so they will be destroyed first. After their activity, they will be destroyed. And if there is no question of hemostasis, no cut, so it will live for the whole, whatever the day it has to spend, it will survive for that period. These are found only in bone marrow. So this is another point. These are formed only in bone marrow, and that too from megakaryocyte. Mega means big. Karyo means nucleus. The big cells, mega cells that are present in bone marrow, no other cells bigger than this. So this is the megakaryocytes, from they originate. And one megakaryocyte can produce 2K to 4K platelets, means 2000 to 4000 platelets. How they form it? Very nice formation. So it is a big cell, and there is cytoplasm and cell membrane. So some part of the membrane is pinched. So some membrane will be there, and within this membrane some part of cytoplasm will be there. So then now this part is teared off, and this is thrombocytes. So in this way, throughout its periphery, continuously, and ultimately the thrombocyte will die. It's like spider, no? When ultimately the mother dies after, when the spiderlets, these are formed, no? So mother is died. And normal blood count of thrombocytes, how much? 1.5 to 4 lakhs. Okay? You have to remember, if their production is raised more than this, what will be the term? Thrombocytopenia or cis. Cytosis means increased production. Penia means less production. So, one is thrombocytosis, another is thrombocytopenia. They will die once. What will happen to them? So, vulture is there. So, vulture means macrophages. So, they will pick up and destroy and fragment it. This is the electron microscopic structure. And this is it's a schematic. Diagrammatic, not the actual. So this is drawn, this is shown to know what are visualize what are the actual constituents. So this is the cell membrane and these are the canaliculi. The cell membrane invisinates within the cell. These are called canaliculi. And within the cell, except nucleus, you will find other organelles. You will get mitochondria, though a few. You will get endoplasmic reticulum. Some proteins are to be formed. Some phospholipids are to be formed. Some glycolipids are to be formed. And these are mainly smooth endoplasmic reticulum. Beside this, what you will get? You will get some contractile elements. These are actin, myosin. These are also present in your muscles, in your smooth muscles. Okay? So, when they contract, these are only the contractile tissues in our body. So, when they contract, the thrombocytes will also contract. So, there is some purpose. So, it will be performing some functions with these contractile proteins. What are these? What are these contractile proteins, as I said? Actin and myosin. Beside this, you will get some granules. These granules are within the vesicle. These granules are of two types. Dense granules and alpha granules. Let's see what is there in these granules, in the dense granules, in the alpha granules. We'll see. The dense body. Platelet cytoplasm contains, these are the factors. What are these? Cytoplasm within the cell membrane, inside the cell membrane. Actin and myosin function, contraction, make platelets spherical and produce pseudopodia, because they are also having some phagocytic activity. They will come. They will be having also some phagocytic activity. Moreover, these pseudopods are also essential for binding other platelets so that they can come closer. Understood? Beside actin and myosin, there is another protein, thrombostatin. What is thrombostatin? Right? Thrombostatin is another contractile protein which can cause platelet to contract. We know after clotting mechanism, blood has been clotted. Now after few minutes, this clot will retract and it will produce serum. Serum, the fluid we get after clotting, this is called serum. So this thrombocytes will help in producing serum by contracting the thrombocyte present in the thrombocytes. Okay? As I said, there will be endoplasmic reticulum, Golgi apparatus. So actually, whatever the secretions, they are first coming into the vesicles, then through the vesicles they are coming out. And some granules will not be within the vesicle, they are free, so they can diffuse out. Or if the membrane is ruptured, they can come out. So, mitochondria is there. What is the function of the mitochondria? Because it has to do so many functions for this energy is required. Since it will not be living for long, so that's why number of mitochondria is relatively less. There is microvesicles, small vesicles we have seen, and microtubules. These microtubules are cytoskeleton, giving the shape to the cell. And glycogen. What the mitochondria will do? If glycogen is not there, glycogen will be broken down into glucose, it will be then oxidized, and pyruvic acid will be formed. This pyruvic acid will be oxidized in the TCA cycle and energy will be produced. So that's why mitochondria is needed. And lysosomes, as I said, they are having the phagocytic activity. When they are phagocyting something within their cell, what they will do then? This phagocytosed material will be added with the lysosomal enzymes, so that the enzymes can destroy the contents, can digest it. Okay. Then granules. We said, no, these are very important. There are some granules. One is dense granule. Underline. Point to be noted. These are granules. There are two dense granules and alpha granules. What is there in these granules? The dense granules contain what? Glycolipids, phospholipid, cholesterol. First thing, we know for clotting mechanism, several factors are required. 12-13 factors are required, and some of these factors, rather most of these factors are synthesized from these. All these factors are actually their function. Function of the platelets is also to produce the coagulation factors. So these will be synthesized from these sources. For that reason, they are present within the cell. And there is 5-hydroxytryptamine and serotonin. They are vasoconstrictor. When these will be released and when they will be coming in contact with the smooth muscles, it will cause contraction of the smooth muscle locally. Means, suppose this is the blood vessel. Suppose this is the blood vessels. Here is the injury. So platelets will try to come out. So these platelets will release locally these contractile substances. What are these? 5-hydroxytryptamine, serotonin. So they will immediately cause constriction of the smooth muscles. So lumen will be narrowed on both sides. So neither then it will be difficult to come, blood to pass through, since it is narrow, so bleeding will be lowering. Moreover, by this time another thing is formed, so there it will cause total blockage. Okay, so no more hemorrhage will be there. So they cause. So these are important, no? You could understand now. So these are also important for producing local vasoconstrictions. These smooth muscles around the small arteries, arteriole, not only they are responsive to 5-hydroxytryptamine or serotonin, they are also under the control of my brain. We know we have two nervous systems, peripheral nervous system. What are these? Sympathetic nervous system and parasympathetic nervous system. Their functions are, on an average you can tell, just opposite. We balance our activity, say activity of heart. When we need more beating of heart, then we activate the sympathetic nervous system. So sympathetic nervous system from its nerve ending will produce adrenaline, epinephrine, and this adrenaline, epinephrine will increase the heart rate. It will, the purpose is to supply more blood. So it will increase the cardiac output. It will increase the stroke volume. The amount of blood coming, oozing out from the heart is called stroke volume per beat. And in 1 minute, it is the minute volume or cardiac output. Okay? So this sympathetic nervous system will also, they can also contract, cause contraction of the smooth muscles. But that contraction will be not locally, but throughout the body. So action of epinephrine, adrenaline is not local. It is as a whole. So there will be increased blood pressure. But when parasympathetic stimulation is there, it will liberate acetylcholine. It will produce vasodilatation. So blood pressure will fall. So with this sympathetic and parasympathetic activity, heartbeat, cardiac activity, we control our blood pressure. Another important thing is their ADP. Here when there is increased breakdown of ATP within the cell, so there will be formation of ADP. When the cell ruptures or cell, it is needed, cell releases this ADP, and ADP causes aggregation of platelets in the injured area. So that is why also ATP very important. And ATP is needed. If ATP is not there, where from ATP will come? So it is obviously, and it has to perform so many functions. So there should be some ATP. So what are the contents of dense granules? Tell, without looking at the slide. Tell, what are the contents? Remember, by heart, because question may come. Phospholipid, glycolipid, cholesterol, because these are needed for clotting factors, production of clotting factors. And this 5-hydroxytryptamine, serotonin, they will cause constriction of blood vessels, local blood vessels, so that to minimize hemorrhage. Okay, then ADP, it will facilitate accumulation of platelets in the injured area, cut area. And ATP stores, as I said, now the alpha granules, they contain, see, these are the precursor of this. From this, these are produced. From dense particles, transgranules, these clotting factors are synthesized. Platelet derived growth factor. So when there is an injury, thrombus will be formed, hemorrhage is stopped, then what will happen to this injured blood vessel? Again it will recover. Again it will regenerate. How? Because these are tissues, there should be mitotic divisions within the cells. So there will be rapid mitotic divisions, whatever the cells are, there they will be dividing so that it can heal. The injured vessel can, and this is done with this platelet derived growth factor. What is the function of platelet growth factor in thrombocyte? Now let us see what is there on the cell membrane. On the cell membrane, we will find receptors. We know hormones act on their target cells by binding. Suppose this is the target, I am the target cells. So I should have some receptor to recognize. I need epinephrine. So this receptor is specific for epinephrine. So where epinephrine, you also look for this receptor. I am also looking for epinephrine. So whenever we will find, we will attach with the receptors. So definitely the cell wall of thrombocytes, platelets, are, as they are having some receptors, they will bind with something. And the purpose is there. So what are these? These are collagen receptors, fibrinogen receptor. And remember this, very important. Why short motion comes? What happens when this factor, VWF factor, is absent in platelet? Okay? What is its function? Okay, that's why it is important. It is this, what is the spelling? What will be the spelling? Von Willebrand factor. This is actually this factor. VWF is a large circulating molecule produced by the endothelium. Whom should I ask? Where you will find endothelium? One yellow shirt? Yeah, you don't have. Can you know, do you know where you will get endothelium? No. Who can tell? None. Only one. And 1.5. It's like this. You know, confused. See, this is the blood vessel. We will get 3 layers. The outermost layer is tunica adventitia. And the middle layer is muscular layer, smooth muscle layer, circularly arranged. I will ask you next day. What I will ask? Where you will get endothelial cells? Why their inner surface is smooth? So, tunica adventitia, outermost layer. Tunica media, made up of smooth muscles. And the inner coat is tunica intima. This layer is actually made up of endothelial cells. And the inner surface is smooth. Again, yes. Why the inner surface is smooth? He is able to answer. Then okay, understood. You are able. Can you?
The inner surface of the blood vessel is smooth because tunica intima, it's made up of endothelium.
Made up of? This is not the answer. Double A na?
Because the tunica media has smooth muscles and it is overlapping the tunica intima, that's why the inner surface is smooth.
See, blood is flowing through the lumen. Okay? If we know that thrombocytes very is fragile, when there is any rough surface, it will be broken. And once broken, the process of clotting will be start within the cell, within the blood vessel. So to prevent this, the inner wall has to be. Who is not listening? Question coming. Question coming. Alert, be alert. Next question coming to you. So, now you think about, if cholesterol is deposited within these epithelial cells, means endothelial cells, they will grow. If the fats are deposited within these, they will grow. Are there any space in this side? No. But they can grow towards the lumen. So, because of growing inside the lumen, towards the lumen, the surface will be irregular. So, when blood will be passing through this and the platelets will be coming in contact with this rough surface, they will be broken. And the process of coagulation will start there. It happens in cardiac attack. It happens in stroke. In cerebral arteries, cerebral branches of cerebral arteries. Clear? I will ask this question another day. So these are the receptors we get.
Sir, so what is the why the layer is why the layer is
Smooth to avoid friction.
how it gets smooth?
The epithelium, single layer epithelium, epithelial cells, only small gap remains. That too in the capillaries, not here. So they are almost tightly, not tightly, they are adhered to one another to make the surface. And we know there are some tissues, there are some factors, they protect these epithelial cells. And these protective agents are there. So their function is to make it smooth, make the layer intact, so that outer organism cannot enter and break it. So, there are some other thing in the membrane. What are this? The membrane contains precursor of thromboxane alpha A2. It is also a vasoconstrictor, plus it also helps aggregation of platelets. Local vasoconstrictor as well as aggregation of platelets. So it is a powerful vasoconstrictor. The membrane contains phospholipid. I talked about this earlier because from this phospholipid some coagulation factors will be formed. This is a diagrammatic view. This is the platelets and platelet glycoprotein. So they are having a receptor. With this receptor, it can bind fibrinogen. Fibrinogen are thread-like structure. Normally they are soluble and inactive. When they become activated, they form fibrin. This fibrin forms a network, but this network is soluble. Then this, with a factor of coagulation, it becomes insoluble. So when it becomes insoluble, you will find at the injured area a network, a net. And within this net, the blood cells are trapped and form the clot. So this is very important. And see, what is this? This is collagen. You will get in the endothelial layer, in the connective tissue layer, the outermost layer. The outermost layer, there is collagen. And just below, just above, towards the exterior, just above the endothelial layer, there are some collagen. When there is injury, this collagen will be opened, tissues will be opened, the outer layer will also be opened, inner smooth muscle will be opened, and endothelial layer will also be opened. Okay, so collagen is very important for formation of platelet aggregation, platelet plaques. This collagen requires one receptor. Without receptor, the platelets cannot bind with the collagen. We know they are having this surface is having three types of receptor: collagen for collagen, and form what is it? Willebrand factor. So that receptor will be also there. So collagen will first bind with this receptor. So through this receptor, collagen can bind with thrombocytes. Now the picture starts. What is the picture? Okay, I'll come later. So this is this is the megakaryocyte. See, the megakaryocyte, its diameter is 35 to 160 microliter. Okay, and see, from its cytoplasm, these are actually platelets. These are actually platelets are forming. Megakaryocyte, large cells, these are small cells. See, these are thrombocytes, platelets are formed by pinching. Okay. And how thrombocytosis, genesis, leukopoiesis, thrombopoiesis. How thrombopoiesis takes place. This is what PAs means pluripotent hemopoietic stem cells. From these stem cells, this is the line. From these stem cells, this megakaryocyte is formed. From this megakaryocyte, platelets are formed. What are the steps? So these are the steps. Okay. Common myeloid progenitor cells, then megakaryoblast, then pro-megakaryocyte, then megakaryocyte, then platelets. These are the steps. The functions. What is the number one function? It is written there, hemostasis. The whole hemostasis process is not done by this. The initial part is initiation of hemostasis actually start with platelets. And it does it through total process of hemostasis takes place through some stages. What are these stages? So if there is a cut, immediately that local area has to be constricted to minimize the flow. Okay? And it is done by local reflexes. Moreover, there are pain receptors. Pain. So this pain will go to the brain. Okay? It will go to the brain. The central nervous system then will stimulate sympathetic nervous system. From the nerve terminal of sympathetic neuron, epinephrine will be liberated and they will cause constriction of the muscles. So by this way, there will be vasoconstriction. So there will be reduction in hemorrhages. So first process is vasoconstriction. It is done a spasm induced by serotonin, 5-hydroxytryptamine and thrombocyte A2 and by reflex vasoconstriction. How it is been done? It is not done alone. So there are some cofactors. These means the serotonin will be involved, 5-hydroxytryptamine will also be involved, thrombocyte A2 will also be involved and reflex vasoconstriction, that will also be involved. And next part is platelet adhesion, then is platelet aggregation, then formation of platelet plug, then only formation of clot. Plug and clot, these are different. Formation of plug is different from clot, formation of clot. Clot formation is later part. And plug formation is the initial, before this it happens. So whenever there is a cut, so blood stops coming out and it is initially because of platelet aggregation, formation of plug, not because of clot. Understood? Vasoconstriction and in the injured area there will be plug formation. That will prevent further oozing of blood. And thereafter, this plug will be retracted. RBCs will be added. There will be the role of so many plasma protein factor, coagulation factors. They will be involved within this process as well as that the clot will be formed. And thereafter, it will be hardened. Then it will be dissolute. And by this time, repairing process has started. And after this, repair will be done. So these are the processes involved. So next function of thrombocytes, it helps in coagulation. Because it produces so many coagulation factors. So thereby it participates in coagulation process by producing so many factors. And clot retraction. We mentioned there are some factors that cause Beside actin and myosin, there is another component. So this component also involved in clot retraction. Phagocytic function, as I said, it has got phagocytic function also. So here, what is phagocytosis? There is phagocytosis of carbon particles and virus and immune complexes. So they engulf it, they can destroy it. Storage and transport. So within the cell, they store this vasoconstrictor. When is needed, it liberates. When it is needed, when the blood vessels is injured, then only it is needed. It is secreted. And repair of injured blood vessels. So now tell me, what are the functions of plasma protein? Number one, hemostasis. Number two, clot formation. Number three, right. Number four, okay. Number five, wow, fine. You learned what are the functions of plasma protein. No need of studying at home. This is the definition of hemostasis. What is the definition? It is the process of forming clots, not the formation of platelet plug. It is the clot, because after the formation of aggregation of platelets, thereafter it happens. It's the second process. These are the events. First, vascular constriction, and then formation of platelets. Today's discussion is limited to this only. And the next discussion will be the later part, clotting mechanism. It is already been discussed. So there is first platelet adhesion, then platelet activation, platelet aggregation, then for there is temporary hemostatic plug. This is the steps. Okay. Then see. Collagen. Now they have receptors. To this receptor collagen is binding, and there are other this receptor. With these receptors also they can bind. So after binding of this, it opens another, the thrombocytes opens fibrin receptors. So with this there is fibrin or fibrinogen in the plasma protein. So they will will attach to the receptor, and on the other end they will one end with this platelets, and the other end it will fibrinogen. So this is the fibrinogen. So with the receptor it will bind one thrombocyte here and another. In this way, lot of thrombocytes will be coming together. This is called platelet adhesion. When this platelet adhesion is there, these platelets become activated. It becomes activated. Then from this, some granules come out. ADP comes out. Now ADP invites further. Hello, thrombocytes, come. Be here. So there is aggregation of platelets. And where they are aggregated, see their location. Here they are aggregated. So there will be no oozing of blood from blood to the surface outside the area, cut area. This is actually hemostasis, clotting mechanism. So this is the aggregation. This part is the aggregation of platelets. Okay. And now in details. So what is happening? Injury, collagen is coming out, and platelets are coming and joining the collagen. Okay. Now These are activated, platelets are activated, pseudopods are formed, ADPs are released, so more platelets are joining and forming the plaque. Let's see in details how it happens. So these are the collagen cells. What are these? Collagen binding receptors. Now with this receptor, with this fibrin, these thrombocytes are joining with the collagen. So they will change the shape like pseudopods. Now they are activated. And now fibrin will come. See, there is the formation of fibrin receptors. Fibrin receptors are coming out. Different factors, this has come already. IDP entered into the picture. So it will attract more lymphocytes. Sorry, not lymphocytes, thrombocytes. So alpha granules release their contents. Fibrin. One end there is one end, there is on the other end there is another thrombocytes. So in this way plaque will be formed. Those who are having probably this is from AKJ, you go through this the overall process. Injury, serotonin secretion, hydroxytryptamine, iron cellulose secretion, vasoconstriction, activation of this, formation of platelet plaques, and then the next part, blood clotting, and variation, thrombocytosis, thrombocytopenia. What are the conditions? Thrombocytosis, increased production. When spleen is removed, when it is removed, spleen, when there is lot of viral activity or there is activity of foreign bodies, then it is the main function of protecting the body from it is actually performed by the spleen. So their activity is increased, and as a result the size is increased. It is called splenomegaly. When there is splenomegaly, spleen lies just above the kidney. So it gives a lot of discomfort. Then doctor will advise remove the spleen. It is called splenectomy. So when splenectomy is there, actually say 100 thrombocytes are formed. 75 will be remain within the circulation and 25 will be contained within the stored within the spleen. So when there is no spleen, so no storage, the count of this in the blood will be increased. But there is problem because spleen is performing some important functions in our body. Okay, so removal of this splenectomy, any kind of stress, it will produce epinephrine, adrenaline, and that will cause contraction of the spleen under normal condition. So it will expel, there will be expulsion of the stored thrombocytes into the circulation. So there will be, count will be raised. And trauma, any type of surgery, trauma, injury, accident, this will also increase the count. And thrombocytopenia. Thrombocytopenia, purpura. You know purpura? Blue coloration of the skin means internal hemorrhage in the skin. So purpura, there is continuous bleeding. So in purpura, it is reduced. Bone marrow depression, aplastic bone, because of X-ray irradiation and some other things, X-ray, gamma ray, and hypersplenism, hyperactivity of spleen. This will store more thrombocytes. So blood levels will be reduced. And viral infection like dengue, this will also reduce platelet. And drug hypersensitivity, there are some drug which causes hypersensitivity. In this case, it will be reduced.